if you want to ask questions or make comments, you're really welcome, and you can interrupt me. And if you don't want to interrupt me and you want to put it in the chat, then Sarah will bring it to my attention as we go along. So kind of I put a question mark here in non-small cell lung cancer because, these biomarkers are now increasingly becoming pan-cancer. And the story started in non-small cell lung cancer, but it's kind of evolving, and I can see that this will become staple diet on many tumor sites. So today we have a brief run through on kind of a background, kind of knowledge that everybody knows, but it's a good reminder on these two molecules, and also on how we go about assessing them. And this is just a few slides to say that the introduction of targeted therapy here, the early one with gefitinib, has marked a transition of oncology where there were tiny differences in improvement in therapy. This is 1976, and this is overall survival. And introduction of platinum doublet, like 25 years later, just made a tiny bit of difference. And now the first tyrosine kinase inhibitor, look what a jump. And now we're kind of moving towards 20 or 30 or 40% survival. And this is the quantum difference that everybody's excited about. And ALK and ROS fit into this conversation. So this is just a slide again to remind you, these are just the various mutation and distribution of mutations within normal cell lung cancer. But look, different quantities in former or current smokers and never smokers. And I'm pretty sure if you subclassify these tumors for other factors, they will also be different proportion. But look how many ALK rearrangement there are in the never smokers compared to the few percentages in those that have chronically exposed to trauma, such as smoking. So ALK, those of us who are relatively old remember the anaplastic lymphoma kinase and this fantastic molecule that emerged where we could certainly identify a subtype of lymphoma because it was the only thing that was positive for anaplastic lymphoma kinase. And there is a story behind it, which is the translocation. And we only really understood the translocation because this was a molecule that was only present in these tumors. And that's a picture of the original clone, ALK1. And I put this on purpose because ALK1 had a specific use in the setting of lymphoma. But it really is not cutting the cake in the setting of other solid tumors. And the reason why is twofold. One is because it's a much better stainer of the translocation that is prevalent in lymphoma, anaplastic lymphoma, but also because its sensitivity is much lower than the current tools we have. So the key fact of this discovery was that this was a chimeric protein. So chimeric is a fusion between two parts that don't really exist together in nature. So it's a chimeric. You remember the chimera are this mythical monster that has a part of one animal and a part of another animal. And this uniqueness made it very useful diagnostically, and only later it became a target of therapy. So the translocation is between a receptor tyrosine kinase and something else. And the something else can vary. And I will tell you that there is a common thread about what is the something else and why. This is kind of 25-year-old knowledge now. And this is the gene that encodes for the anaplastic lymphoma kinase. The anaplastic lymphoma kinase is a full-length transmembrane tyrosine kinase receptor. it's kind of big. You see 26, 29 exons, etc. And the protein is quite big, 180 kilodalton. This is the native protein, not the chimeric protein resulting in fusion. And the important part of this model, so that's the kind of extracellular domains. This is transmembrane domain, and this is the tyrosine kinase domain. And the important part of this molecule for our purpose is the tyrosine kinase domain. This is an intracellular portion. And as it often happens in all these receptor tyrosine kinases, the tyrosine kinase gets activated because two of these receptors are just opposed very closely, and the intracellular portion of the molecule, the tyrosine kinase domain, becomes close to each other, and it becomes activated, and then it can phosphorylate and do whatever it is signaling that it does. And all these are kind of information that are interesting but irrelevant. So it dimerizes. This is the concept. It needs to dimerize in order to be activated. That is normal function. And it is called proto-oncogene. And if you remember our kind of developmental cellular biology, a proto-oncogene is a gene that is a normal gene, but can induce tumor in certain circumstances. It becomes an oncogene when mutated, or sometimes overexpression, because overexpression is usually the result of amplification. And amplification is a form, usually is a signaling molecule in the central nervous system. There are very weak labeling of normal tissue in various circumstances. And this is why we use nervous system cells for our on-slide control, because they have much better staining. So that's the kind of a tyrosine kinase receptor. This is intracellular portion. That's the cell membrane. That's the outside environment. And this is the ALK ligand that binds to the receptor. Let me forward it. When it binds to the receptor, it exposes a dimerization domain, and then two dimers get together. There is proximity, and the proximity results into phosphorylation. And then this is now an active molecule, and there are a lot of kind of intracellular messengers and bridges, and there is a cascade. And these are the usual cascade that everybody is very familiar. One of it is RAS, RAF, MEK, and ERK. Another one is the PI3 kinase, ALK, AKT, axis, etc., etc. So, and then eventually this results into signaling into the nucleus and some actions related to survival, apoptosis, proliferation, motility, etc., etc. Okay, so that's the kind of great scheme of things. So you can understand how signaling up here can become an oncogenic signal for some tumors. And this is some consideration. The consideration is that this chimeric protein that becomes an oncogenic driver is present in a lot of diseases. And here are annotated, well, this was the original index disease, anaplastic cell lymphoma. But for instance, inflammatory myelofibrotic tumor, about 50% have this mutation. And in this context, it becomes a very useful molecule, because I can do an ALK staining, and if it's positive, I can make my diagnosis. And spitzoid or Spitz tumors are also rich in this translocation. This is your non-small cell lung cancer. And remember, it depends whether this is a cohort of patients that are never smokers or have been exposed to chronic irritation. But it appears at low level in many other diseases. And the partner of the fusion can vary. And these are the common partners, and some of the partners are common in some of these diseases, and is annotated here. But in reality, the number of fusion partners is very, very large, but they occur at very low frequencies. So how does the actual how does this this occur? So one of the most common partners of fusion is EML4, and in this case is a little inversion. So the DNA is cut at these points. This is where the ALK gene is, and this is the MLA4 gene, and it kind of switched around and fused in this way. So you create two novel signals. One is ALK and MLA4, and the other one is MLA4 and ALK. Well, this one doesn't really do anything, but this one here actually is a very effective functional fusion protein. And the key of all these partners is that they have this domain that is fused to ALK, is a domain that enables self-assemble, which means that you don't need a, um, a ligand for this tyrosine kinase receptor, because the tyrosine kinase receptor assembles itself into a dimer because the EML4 partner is coiling with each other. So the coils spontaneous dimerization and therefore activation of the ALK molecule, as is shown here. So in normal cell lung cancer, there are preferred partners, and they're indicated here. Some of them are within the same chromosome, some are in other chromosomes, so the translocation rather than inversions. But in fact, I was telling you that now that there is sequencing, so there is a much wider range of molecules associated with this fusion, and this is the last publication were 92, so the potential partners are quite large, although they occur in very low frequencies. So the other type of ALK mutation that is relevant in oncogenesis is point mutation. And this is not dissimilar to a lot of other tyrosine kinase receptors where there are point mutations affecting the kinase domain. Essentially, the kinase domain doesn't need activation, dimerization, because the point mutation makes it constitutively active. We are more often in particular diseases. For instance, in neuroblastoma, there is a classical point mutation. Most of these occur secondary because of therapy that induces this type of mutation. There is a selection pressure. Patients are treated with these tyrosine kinase inhibitors, and the cells develop mutations that bypass this mechanism. And here are a list of point mutations that are associated with resistance to soluble tyrosine kinase mutation. Now, the third thing we should consider is amplification, gene amplification. Why? Because it does happen, but most often actually is amplification of the fusion genes, not of the native genes, but of the fusion genes. And okay, so let me just close this a little bit. Okay, so we were kind of going through how the ALK molecule fires. So this is the normal scheme of things when there is a ligand, the dimerization, and the downstream signaling. So that's normal. And then this is when there is a translocation. The molecule doesn't need to be in the membrane, doesn't need a ligand. The fusion partner, dimerize the molecule, and the tyrosine kinase can work downstream. And this is the mutant ALK where the tyrosine kinase receptor is activated regardless of dimerization. And that means that it doesn't need a ligand, and it can activate downstream pathways. And then the fourth one is if you actually express a lot of ALK, regardless of whether some of them are activated and dimerized, you flood the membrane and you could have a ligand-dependent constitutive activation. So there are circumstances where the normal molecule is expressed in such a high amounts that it becomes activated by force. These are much more rare circumstances. This is the major occurrence in non-small cell lung cancer. And these are lists of these small molecule inhibitors, soluble drugs that um, that interfere with the pocket that binds the tyrosine. So in the tyrosine kinase receptor, in this enzyme, there are binding pockets, and some of these small molecules have a much higher affinity for the enzyme than its normal ligand, and therefore they stop tyrosine kinase activity. And the effect, these were some of the original paper, the effect on tumor was amazing. This is a young patient with advanced non-small cell lung cancer presentation, and 2 weeks after treatment, you can see the resolution of the lesions. The tumor melts away. Typically, these patients go from not being able to breathe normally and essentially being bedridden and having about 2 to 3 weeks of life to go home and have their normal activity, including their physical running, etc., within 4 weeks of treatment. So these are amazing drugs. And are these small molecule inhibitors. Crizotinib was the prototype. You see, they're very small molecules. They sit in the pocket, the ATP binding pocket. They don't let go of it, and therefore they stop the tyrosine kinase working. And of course, as the time progresses, pharmaceutical companies have come up with better small molecule inhibitors. The reality of life is that in tumors, let's say in small cell lung cancer, there is an excellent response. Not all patients respond, but a large proportion of these patients responds. But then sooner or later there is a resistance, and then the tumor progresses. And the second generation and the third generation of tyrosine kinase inhibitors are indicated here. They overcome some of the resistance mechanisms, and they provide longer lasting responses. So what are the mechanism of resistance? There are various mechanisms. One is the on-target mechanism of resistance, and we discussed this before. There are mutation in the tyrosine kinase binding sites, and these represent about about a quarter of the resistance mechanism. And there are the off targets. So basically the cell learns to develop a bypassing signaling strategy for this signaling pathway. So they use another pathway, and it doesn't really mind that the ALK activation is switched off. And how do we test? Maybe now things are becoming more interesting for pathologists. So how do we test for ALK? We kind of need cancer cells, or we could use circulating cell-free tumor DNA. So that's a peripheral blood job, and we are going to sequence to see whether there is a translocated signal molecule there. And you remember these are non-existing DNA fragments. They are only created by translocation, so that is a unique fingerprint. And instead, if you can get access to cancer cell, which with whatever way you want, then you can measure it on the cells. Remember that if you have a biopsy from a metastatic side or rib, etc., remember to fix well, and then do not use formic acid to decalcify. But the most important thing is fix well and not be in a hurry to decalcify tissue. All the other things, especially the cytology samples, avoid fixing in alcohol. So the first fixative of choice in order to use the downstream test that we use for this test, for this molecule, is fixing formalin. If you fix in alcohol is a fixative that precipitate proteins, and therefore the antigenic recognition of alcohol fixed material is much poorer. Okay, and then so how do we test for ALK? So we can do in situ hybridization. This has always been considered in the past the gold standard, but maybe we will see why this is not entirely true anymore. Immunohistochemistry, which is the technique more close to our heart. We can use PCR technology, perhaps a reverse transcribe PCR. So you take the message that contained the chimeric gene, and then you reverse transcribe, you make a cDNA, and then you PCR that, or you do sequencing using various platforms. So this was the gold standard. In the past, it was the only standard. Then it became the gold standard when immunohistochemistry was introduced in a more robust fashion. But of course, it is important to have good quality of tissue, and it is an operative intensive technique, and sometime if the inversions are very, very close together, it will not be able to detect. I will show you some pictures on why this is the case. Immunohistochemistry, you need to be aware that some antibodies are not sufficiently sensitive to detect this molecule, and the D5F3 has been now kind of heralded as the gold standard for immunohistochemistry. The results are available much quicker and can be interpreted by us using a light microscope, not a fluorescent microscope, and possibly is much more accessible to patients because immunohistochemistry is more accessible than FISH. Remember that there will be cases, and there will be cases where all these technologies concur, but a few cases where some of these platforms are not indicating that there is a mutation. And in particular, we are very concerned or very mindful of those cases where the immunohistochemistry is positive, but the in situ hybridization is negative, because it has been shown that these patients actually are responders. And in fact, the converse, those that can be shown to have translocation by in situ hybridization, but no protein expression, these are non-responders. And that's clearly because the target of the drug is the protein, is not the DNA. Let me see if I have. This is an example of a patient that was that had translocation negative, FISH negative, it says here, but antibody positive, and there is good response in few weeks. Let me just show you break apart FISH. This is how it is assessed. So normally the ALK probes are close together, and in fact they are almost fusing and produce this yellow color. And if the ALK molecule is broken and part of it is physically in another place in the genome, you have two different dots. Now this work very well, except where the translocation is very, very close to the ALK signal. You will still see two dots together, and you will not detect it very easily by FISH. And there is a convention that looking at so many cells, calculate the percentage of rearranged cells, and then have a threshold that indicate positivity. So that's convention. So if you're interested in measuring ALK by protein, I think this is the Bible, which I think is freely available. And there is a good understanding of all these technologies, including in situ hybridization, and a detailed understanding of what does the immunohistochemistry look like in these translocated cases. And this is a list of antibodies that could be used. We spoke about some of the antibodies that are not very sensitive, and I think everybody now is kind of concentrating on the antibodies that have shown to have very high sensitivity and specificity for responses to therapy. Very briefly, whenever you do immunohistochemistry now, it's very important to have an understanding whether your slide has been exposed to the right chemicals, and use of on-slide control is very important. Typically, you can use cell lines if you want to, with translocated or not, or overexpressor or non-expressor cells. But actually, we find that appendix which is freely available in many histopathology departments, is a very good source of control. These are the neuronal cells of the Auerbach plexus, and if I don't know if I have a higher power picture, they stain quite strongly. Remember, this is a molecule expressed in the nervous system. No, I don't have a higher power pointer, but with correct immunohistochemistry, you will find that there are the small nerve fibers. You will see the small nerve fibers, individual nerve fibers in the mucosa. And that gives you an understanding the sensitivity is good. And of course, this has got a lot of non-staining cells, negative cells. You will already see at a glance that the mucus may retain signals. So you need to watch out of these things. These are some examples of staining. This is probably an effusion because it looks like these are inverted cell balls. And you can see here with the antibody, it's very clear when you have a positive result. This is the on-slide control. That's a slightly high power. And you can see here the negatively stained nucleus. The signal is more often than not in cytoplasm, but actually the distribution of staining and the quality of the staining, whether it's granular, whether it's coarse, whether it's linear, has some relationship to the fusion partners. So some people who are very good at interpreting immunohistochemistry will also tell you the potential fusion partners of the molecule based on the immunohistochemical appearances. Okay. So I think I'll skip that because already we lost a little bit of time, but I wanted to discuss with you workflow and management for diagnostic lung biopsies because that's important. And we often say that we've often been met by the problem that lung biopsies, EBAs, etc., are very small and we run out of tissue before we can do all the biomarkers required. That's not true. Once you tightly control the workflow in the laboratory, you realize that there is enough tissue to do everything. And that's what we normally do in the laboratory. So any cytology is not smeared. That's a waste of cells. All cytology is created, is spun down and a cell block is made. And then the block is faced. We don't throw anything. So all the curls that come out of the facing of the block are saved. Then you cut the spare section ahead of time. The reason why you do that is that if you then later on, tomorrow, the day after tomorrow, you put the block back in the microtome, it requires facing again. So you throw away a bit of sections again. So you cut and save a lot of spares. Then you look at the H&E and make your own assessment, diagnostic assessment. You may want to order some immuno, whatever it is. The technologist has already cut spares. And then once you make the primary diagnosis, you request all the on-slide biomarker that you need. In this case, for instance, here is ALK, ROS, BRAF, PD-L1, and NTRK. And then you look at the slide report. If you don't need additional tests associated with these slides, and then you take the additional material and you request EGFR, or if you want, you request FISH if you want to resolve some problem with ROS or NTRK. And there is still enough tissue in the block for you to do it, but in any case, there are also tissue saved in the cells when we face it for DNA extraction. So this kind of workflow will give tissue for everybody. In many years of practice, we essentially have very few, less than a full hand of cases where we could not get enough DNA for the EGFR tests. And of course, if you want, you can do some downstream NGS studies. Okay, if run through the ALK situation. And I wanted to use the rest of the time to look at ROS. Let's put, and Sarah, please do tell me if there are questions or if anybody wants to unmute and just make some comments, please do that.
There are no questions.
Okay, so
Carry on.
cool. Okay, so again, kind of sorry, this is a bit of a summary slide that say there are all these mutations, and there are is an increasing list of mutations, rearrangement, etc., that we are expected to deliver to the oncologist in order for a patient to be treated effectively. Um, there are now targetable, this slide is older, so now there are targetable KRAS mutation, particularly the G12C and G12D. Uh, uh, and I think next year we'll see targeted PTEN loss of function. We are already in the space of treating PI3 kinase mutation. So all of this is a highly emerging, evolving system. You remember now that the dual antibody drug conjugate Enhertu is also in second line for HER2 amplified um, non-small cell lung cancer. So these are all new targeted biological therapy for these patients. Okay, something is missing here. I don't know, but until we'll skip this for a moment. Sorry. And I wanted to tell you that so ROS1 is a kind of the same story as ALK, is another one of these tyrosine kinase receptors that is the focus of a rearrangement, and then there is a fusion protein, and then you can target the fusion protein because it's oncogenic, and then you have a fantastic response. But this one is an amazing story because from discovery to actual phase 2 published trials is the shortest time frame for anything that developed. And the reason for it is that the trials were so clear-cut responses that immediately the drug went on approval. And this was a pre-1994 number of publications, and now, it's arrived. Everybody's excited about this. Okay, so that's one of the cases, one of the early cases. You can see this baseline metastasis everywhere. 7 weeks after on crizotinib, this is the same drug that was effective for ALK. These are early days. We are in 2014. And this is the classical waterfall plot that shows that there are a lot of complete responses and partial responses, and a few stable disease. This was one of the first trials. Okay, so lots of excitement. What do we know about ROS1? So here is ROS1 is a transmembrane receptor. It has accessory molecules, intracellular, this SHIP1 and SHIP2, and then it activate either the RAS, MEK, and MAP kinase pathways or other pathways. As you can see here, this is the PI3 kinase AKT pathway. Now, interestingly, let me just move this. So interestingly, okay, these are the accessory molecules, and interesting, there have been some areas of pathology where mutation in these proteins are part of a certain syndrome, but that's not kind of very relevant for us. This is the schematic representation of where the gene is on chromosome 6, and is again a very large gene, is a highly conserved molecule across development, and it is again a proto-oncogene. Where do we find in humans? We can find them very, very, very much. In fact, we all struggle to find normal tissue or even disease tissue that has that we can use as control. Epididymis, cerebral cortex, and lung. Epithelial cells in the lung, involved in maturation, and somehow is involved in gonads maturation. And again, kind of the music you see is very similar to the story of ALK with partners. Here is an example of a swap over between a piece of DNA from chromosome 12 to chromosome 6, resulting in this fusion gene between ROS1 and ERC1. And these are the known partners of ROS1 fusion that result in an oncogenic rearrangement. And the second type of mutation is a point mutation, again, mostly therapy-induced, again, mostly affected in the kinase domain, and these are the point mutations that are known to cause resistance, and these are the fusion proteins where these point mutations are under which this point mutation develops as a resistant mechanism, and then amplification is again another point of another type of mutation. And again, the system is still the same. If I create a very useful gene by a chimeric fusion, then there is pressure, selection pressure, to amplify this gene in the genome. So you find multiple copies of this fusion gene. Where is it present? In a lot of carcinomas and other tumors. Interestingly, schizoid neoplasm are rich in it. Most other tumors have low prevalence. Again, you remember inflammatory myelofibrosis tumor had a lot of ALK mutation. They also have a lot of ROS1 translocations, but low level in a lot of we are currently chasing 0.5 or 1% prevalence in non-small cell lung cancer. I think we will be chasing similar prevalences in a lot of other carcinomas in the future. I've highlighted this here because we have a particular interest in the use of these tools and these antibodies, not only to find the right therapeutic avenue for the patient, but also in providing the right diagnosis. And I think we had a workshop around the fact that these translocation antibodies are useful to identify lineage of differentiation for cutaneous melanocytic neoplasms. And so I've highlighted here the fact that because Spitz tumors or Spitz melanocytoma, now called, are often driven by a translocation, then looking for translocation using antibodies becomes a useful tool when you have a difficult melanocytic lesion and you cannot decide whether this is a classical melanoma or a Spitz-like melanoma, or in fact is a Spitz nevus, originally now called Spitz tumor. Okay, so these are again lists of the oncogenic fusion partners in non-small cell lung cancer, but it's not very important. So how do we screen tumors for this oncogenic fusion? So same strategy, break aparts. So this is normal, and these are apart signals. And there are a number of providers of these tests. And it's based on the fact that the red probe and the green probe are close together in the normal genome, but then if it's a portion of the ROS1 is translocated, the red probe will be somewhere else. And you see here, especially in a distinct portion of the cell, but also by immunohistochemistry. Now, while the immunohistochemistry approach with ALK has been proven really successful, i.e., if I find immunohistochemical evidence of protein expression to the level required, I can treat the patient with confidence that there is an oncogenic driver there. The fact that I find evidence of overexpression, and you can see here, this is a membrane staining, overexpression by immunohistochemistry doesn't necessarily mean that there is a translocation, and therefore doesn't necessarily mean that the tumor will respond to tyrosine kinase inhibitors. So the situation is slightly different, and this could be a screening tool, but if positive, you need to confirm that there is an actionable mutation by a second mean. It could be sequencing, or it could be, if you want, ISH, etc., etc. Interestingly, now that in a lot of places sequencing has become a upfront tool, so instead of doing single gene analysis or single antibody analysis, we just do a panel of 20 or 25 or 40 genes, and we may find sequencing evidence of ROS translocation. Now it becomes important to prove that there is protein expression, because if there is no protein expression, there is no target for the treatment, and therefore that treatment is really not available for the patient. So now the emphasis on people that use sequencing up front is shifted into immunohistochemistry in second line to prove that the molecule is there. And there are a lot of antibodies available, and we use the spring antibody. They're all capable. I mean, this is maybe not as good. This one, there are a lot of publications that show sensitivity and specificity of these antibodies much better, and the spring clone is probably performing slightly better than the 4D6D antibodies. So some don't know what this is. This is the current scheme, the scheme I showed you before. How does the break apart work? It doesn't matter. We don't need it. Again, if you report ROS1, I think these are the publications you really should need. The Atlas. Sorry, I went too far. This is in a way, a good review for testing ROS1. This is the paper that describe as the interpretation of SP384, which is the new clone, and this is another interesting paper that compare immunohistochemistry and in situ hybridization. Okay. We are kind of, I have some more stuff here, but I'm not entirely sure it's hugely important. Okay, so these are comparison between various antibody clones. These are kind of rock curves about specificity and sensitivity. You can see they're very good performers. And I wanted to show you criteria for interpretation. Just a moment. And these are papers. I've given you the reference. These are papers that describe the concordance between ISH and ROS. And these are some examples of staining with the two antibodies. And notably, what you can see is that the previous generation antibody doesn't really manage to stain the normal lung. Here you can see this low level staining in the normal lung. So this is a bit more sensitive, and you can see here as well. I'll skip this because yes, I want to find you these very useful interpretation. So we are back to pathologist domain. We score staining intensity using 3 gears, were 4 really. 0 means nothing, I can't see anything. And this is weak staining, 1+. This is moderate staining, 2+, and this is strong staining intensity. So we are looking for strong staining intensity or moderate staining intensity, and staining and over 30% in order to consider a second test, which is PCR or sequencing or ISH. And this is the evidence based on these type of classifications. Okay. Just really to say that using this two-pronged approach, the false negative and false positive are relatively small in this case. Okay. This is what we do at the moment, but I think again this is a landscape that is changing because sequencing is becoming more and more accessible. We use IHC upfront as a first-line test. If negative, will report as having no oncogenic fusion detected. If positive, we need to reflex to something. We used to reflex to fish, but now I think sequencing is becoming better for this. And then if there is discordancy between these two tests, you should, so if you do use fish, because fish has the problem of not detecting close rearrangements, I think you should go and use an alternative test like PCR or NGS. Okay, so that's kind of the end of my slide deck. And I can stop sharing, and we can open discussion. I can see something in the chat. In the chat, there is a comment about regarding ROS1 interpretation. If we have diffuse but weak staining intensity, should we reflex to ISH or another method? Actually, you can do that, but I think the concordance, you don't find fusions. You can always reflex if you want to. That's not, no, it's not that you mustn't, but in reality you don't find translocation there. This threshold about 30% and staining intensity at least 2+ seems to be quite robust for identifying these cases. The other thing that we didn't say was that the localization of the signal should be cytoplasmic or membrane or both, but probably not nuclear. Okay, some more questions here. In regard to ALK, do you test routinely squamous cell carcinoma, and what is the positivity rate? So I think this is Savelina is asking this question. I think if you remember, originally everybody was told the squamous cell carcinoma doesn't have these mutations. It's a waste of time in searching. Actually, there are two comments on that. Often our samples are very small, so even if we have an understanding that we are dealing with a squamous cell carcinoma, we don't know what the whole tumor looks like. So that's the first thing. So sampling may bias our original diagnosis. But the second thing is that now increasingly also squamous cell carcinoma are reported to have mutations. So yes, we do stain everything. There's another question here. How do we quantify the staining intensity? Do we use the magnification rule? Remember that we use the magnification rule for membrane staining. The thing is this, magnification rule is useful for membrane staining, but is not useful for diffuse staining. So no, I think that's a very good question, to be honest with you. We don't have a rule to define 1+, 2+, and 3+. And this may be a weak link in the interpretation, and maybe we should think about how do we go about it. Eleftherios has a raised hand. Please go ahead.
Yes. Just a comment from our experience. We use the D5F3 antibody, and the interpretation guide, because of the intensity of staining, which is a result of the enhancer that they used in their technique, Roche uses in their technique, is very intense. So the interpretation is yes or no. There are antibodies, I have seen one from DACO, not the ALK one, that their interpretation guide is 1+, 2+, 3+. So some people take that antibody and then they score it as yes or no. And if you go into Nordic QC, external quality assessments, you will see that most of the problems are if you are not following the interpretation guidelines and you are scoring one antibody with another antibody's interpretation guide. So that is what comes out of our experience.
This is a very, very, very useful comment, and I think we kind of used to think of immunohistochemics as being one gear, but now we have a very good concept, and thank you for reminding us that each test is a different test, even though the targets are the same. Yes, absolutely, I agree. So The other interesting thing that you need to remember is that although we use a 30% cutoff, and somebody here asked also what is the 30% cutoff is in ROS1, but what is the cutoff for ALK positivity in for ALK? And the cutoff is one cell. That is, if you have a small biopsy, one cell will trigger. But the thing is this, that normally these are early driver of the oncogenic process. So you expect to see these diffusely expressed rather than restricted in a small proportion of the cells. Although we know there are tumors that are mosaics, and these tumors do respond effectively to treatment. So normally the situation is either all staining or not staining, but a small amount of staining could be relevant, I think. What do you think, Eleftherios? I know you have a lot of experience with the antibody. Do you find diffuse staining or to all cells, or
most of the time diffuse staining? Most of the time all the cells, but there are cases where you don't find. You just find some of the cells that are stained. So yes, we count them as positive, of course.
Yes.
It is very important to trust your online controls when you are doing this testing. This is another comment that I wanted to make. You have to use on-slide controls.
Yes. And do you use appendix or do you use other things?
Appendix.
Yeah. And while we're on slide control, I can give you some tips in general. And we found out that the tissue you process for on slide control really need to be fixed very thoroughly. So we usually do an extra 24-hour fixation before we process it. And then rather than using shortcuts, we use long processing. So we usually use 22 hours or so processing for these things. And that becomes a much more stable block throughout the depth of the tissue. This doesn't happen only in ALK, PD-L1. I think we learned this with PD-L1 really, and we're applying across all the other controls. So excellent. So if there are no more comments, I think there will be now a circulation for ALK and ROS. So there are very few cases, and it's just an occasion to kind of test ourselves against everybody else. I think Sarah will open the circulation shortly after this, and then it will be open for 2 weeks, and then we'll meet again at the feedback meeting to see how we've all done.